So for any of you that were at BIF this year, this will be kind of a very familiar talk, but we have added some new stuff towards the end, so there will be something new for you guys to see. So what I want to talk about is we were interested in looking at reproductive efficiency. Now, reproductive efficiency is an important element of the cow-calf industry because if the female does not produce a calf, she actually becomes a liability. Either cull her or you keep her around feeding her for another year to breed her again the next year. So what we wanted to do was to identify regions of the genome that are associated with reproductive efficiency in beef cattle. Now, there are some challenges when looking at reproductive traits. It's a lowly heritable trait. It's also a complex trait that's influenced by multiple genes. And as a result, this creates difficulty when identifying genomic regions. So we utilized a couple different tools. First was pooling. We took DNA from a number of animals and pooled them into an individual sample. And then we also utilized the latest SNP chip, the 770 HD bovine chip. And just to give you an idea of what we were able to gain from utilizing these two tools, if we were to spend money genotyping each individual animal, the project I'm going to present would cost over $1 million. And by pooling and using the 770 chip, we were actually able to decrease that cost to around $30,000. So it does have also dollar impact. So this is an example of the pooling. This is for the US MARC population that I'll present in the next slide. But we had the low reproductive animals and the high reproductive animals. And so we took 100 animals and put them into each pool. So we had a total of 10 pools totaling 1,000 animals, and then ran these in duplicate on the chip. So to give you a little background of the populations we looked at, the first was a population from right here at US MARC, where we have a fairly diverse population. We have both continental and British breeds. And again, we had 1,000 low reproductive animals and 1,000 high reproductive animals that were split into 10 pools for each phenotype. The next population we looked at was the Deseret Ranch down in Florida. Now, the interesting thing about the Deseret Ranch is that they have a high Bos indicus influence. They have Brangus, Cimbra, and Brayford. And then another interesting thing, which we found very useful, is that they keep their animals around for 2 breeding seasons. So that they don't cull them right away if they become open, they actually keep them around. So we had 2 breeding records. So we had open animals, open pregnant, pregnant open, and pregnant pregnant. The other 2 additional populations that I didn't present at BIF, but I'll show here, is we looked at the Rex Ranch out in Ashby, Nebraska, where we had records from 2007 born heifers, and we have 3 breeding seasons for these heifers. So we have a population of open animals, and then we have a population of animals that were pregnant all 3 times. And then we're also doing some collaborative work with Milt Thomas at New Mexico State University, and he let us have access to his populations, including Camp Cooley, Bassoon, UC Davis, Corona, Juan, Branch, and Circle A. And so we have really a variety of animals that we were looking at to identify regions of interest associated with reproductive efficiency. Now, the next couple of graphs or plots that I'm going to present are going to look like this. We have the chromosomes along the bottom from 1 to 29, and then the sex chromosomes. And anything that's above the green dashed line here is a SNP that was shown to be significant. Now, this is a different graph than the one I presented at BIF. We've tried to do some different analyses, and this is a more liberal analysis. The one I presented at BIF, we only had a couple regions that were significant, 1, 9, and 17, but when we get more liberal with the analysis, as you can see, there's a number of regions across the entire genome that light up. Now, one thing that you'll notice out here at the edge is that we see the Y chromosome. Now, this was kind of a surprise to us because these are all females. They should not have the Y chromosome. That's only present in the males. And so we were really kind of shocked to see that we did have the Y chromosome present, or SNPs on the Y chromosome that were associated with reproductive efficiency. So next, looking at the Deseret population, we were first interested to see if the Y chromosome thing repeated, and it did. Again, in the Deseret population, we see the Y chromosome. We also see other regions across the genome, chromosome 5, which has been shown by other groups to be associated with twinning, and then we have some other chromosomal regions that are significant. Now we wanted to look deeper into this Y chromosome and why we're seeing it in females. And the data here is for the for the Deseret population, where we had the Brangus animals, the Cimbra, and the Brayford. The 00 are the open animals. Then we have the open pregnant, pregnant open, and the pregnant pregnant. And what we saw was that only in the open animals do we see the Y chromosome. So if the animal's pregnant, we do not see it. It's only if they're open we see the Y chromosome. And just to give you an example of comparison, these are two bulls that obviously have the Y chromosome. So it's a fairly high level of Y we're seeing in these pools of open animals. Now we wanted to look at our additional populations. So this is for the Rex Ranch. And again, we have regions that replicate across the genome. We're seeing chromosome 1 again and chromosome 17. And then again, we're seeing our Y chromosome. And this is all of Milt's populations combined. Now we decided to combine all the populations because we only had a small number of pools for each of its populations. But again, just to show you, we are seeing regions that replicate, chromosome 5 again and 11, and we're just seeing a little bit of the Y. And one reason for this is when I talked to Milt, he said that some of these populations do a really good job over the years of selecting for fertility. They put a high emphasis on fertility. And in fact, those ranches that had a high emphasis on fertility were not seeing the Y chromosome. So we were kind of glad to see that if you do put emphasis on reproduction, you do not see the Y, unlike the other populations we did look at. So this is just a figure. There's one graph for each of the populations. The first is the MARC, the Deseret, the Rex, and these are Milt samples. And this is just for the Y chromosome. So we have across the entire chromosome along here, and each of these circles is an individual SNP. And again, we have our green line of significance, and as you can see, across the entire Y chromosome, we are seeing SNPs that are significant. So it's just not one region of the Y chromosome. We're actually seeing it across the Y, indicating that possibly we don't know if it's fragments or it is the entire Y chromosome, but it is significant, multiple SNPs across the entire chromosome. So the first thing we want to determine is why are we seeing these Y SNPs? You know, can we determine which females possess the SNP? And of course, what would it mean to the industry if we could identify females right away before breeding that are not going to get pregnant? So you can automatically put them into the feedlot instead of spending time on them to get pregnant. So what we did was we wanted to evaluate individual females that make up the open pools. And so we utilized a PCR test, and this PCR test is one that I got because it is used to sex embryos. Reproductive departments use it for identifying male embryos versus female embryos. So only a male embryo should give you a positive result. And what we found was I have 12 animals here. They're all females that were in the open pools, and a number of them are positive for this sexing test. And just to give you an example, these are two bulls. So we do have some females that are just as intense as we see in the bulls. In fact, we're able to get 21 to 29% of the animals, or the females in our open pool, were positive for this Y test. So now we wanted to look at SNPs that were specific to the 770 chip. So we identified a number of SNPs that we wanted to look at that were positive for open females on the chip, but not just look at the pools, we wanted to look at the individuals that compose those pools. And so we have 21 animals here, all females, and keep in mind these animals were positive for this test. However, when we look at individual Y SNPs, we see a different pattern. So not all of them are present for the Y SNPs that we evaluated. In fact, we see differing patterns. For example, this animal here is positive for the sexing test, positive for this test, but she might not be positive for another SNP that we look at. So that kind of leads us to believe that maybe there's just fragments of the Y chromosome that are contaminating these females and allowing them not to get pregnant. So one of the things we first thought of when we saw this Y is, are we seeing undetected females? Or, sorry, undetected free martins, not females. Because these females commonly, they calve on pasture, so it is possible that maybe a twin is not being detected. However, when I talked to the Deseret Ranch, they do pre-breed scanning where they do track scores, and any track that looks questionable, that animal gets culled right away. And so they don't think, or they do a very good job of already culling out any free martin that might be there. And then we also do keep good twin records here at US MARC to get rid of any free martins, and so there shouldn't be any of those in our pools that we looked at. However, we still wanted to look at it just to see if it possibly is undetected free martins. So what we did was we took pools of free martins, ran them on the chip, and we saw a similar thing, where we do see Y chromosome or Y SNP in these females. So then we did our sexing test on each individual, and most of them do light up for the sexing test. And again, we had the 2 bulls and 2 cows that are clean. However, unlike our other populations that we looked at where we saw a variation in our sexing test versus individual Y SNP primer tests, the free martins were positive for all of them. If they were positive for the sexing test, they were positive for the Y SNP. And so since we see this difference in free martins versus the open females that were in our pools, again, we think that what we're seeing is not free martin or undetected free martins, it's something else. So conclusions, when we did start this research, we did want to identify autosomes, regions on the autosomes that were associated with reproductive efficiency, and we were able to do that. And probably one of the most important things is we were able to replicate this across multiple populations, which is always important. And then, of course, we were able to identify Y SNP in individuals of open pools that may contribute to the low reproductive efficiency. And depending on what test we use, whether they're specific SNPs on the 770 chip or the sexing test, which we looked at, we're able to detect 3 to 29% of open females in the population. And I'd like to thank all the people that contributed to this work. First of all, the ranches that allowed us access to their phenotypes and their DNA. Bonnie Long, who was very helpful with organizing all the samples and getting them to us. Of course, the 3 statistical individuals who, without this work, it wouldn't have been done, especially John Keel, who's been doing a lot of work with the analysis, Milt Thomas and his lab personnel, and then the 2 individuals that did all the DNA extraction and pooling, Tammy Sorensen and Steve Simcox. And with that, be willing to entertain any questions you might have.